So we had this chapter is about, I'm just saying it over, is about taking food, like the energy from our food, and converting that into ATP. To be more specific, we're taking the electrons off of our food and we're using those electrons to put the P and the ADP back together. Because when you make ATP, that's an anabolic reaction, right? Anabolic reactions build things, but you need energy. So we're building ATP. We're making it from like the P and the ADP. We're going to put it together. So you need energy. Where are we going to get the energy? From our food. So that's what the Krebs cycle does. Pulls all the electrons off of what used to be our food, and we're going to use that to put the ADP and the P back together. So it's anabolic, right? It's kind of both. I mean, it's really catabolic because we're taking your food and breaking it all up until it's nothing. But then we're using that energy to create ATP. So glycolysis, the first step was all about breaking down glucose. We broke it into pyruvate. So 1 glucose, 2 pyruvates, right? Then we, with the pens, there's my molecule of glucose. Glycolysis just went like that, right? Now we have to turn these 2 pyruvates into acetyl-CoA. So you just modify them a little bit, take them out. Now they're acetyl-CoAs, and now they can go into the Krebs cycle. The Krebs cycle, this won't look like this anymore. It's going to get ripped apart. And so now all we're left with is electrons. We're going to take all those electrons over to the electron transport chain. And we're going to use those electrons to power these pumps. So when you look at the membrane, again, we're looking at like this black line in between the brown, the burnt amber, and the canary yellow. That's what this is, right? So there's all these pumps built into the membrane. So you don't see them, but they're like, well, they're built into that black line. You've got these, that's what these purple blobs are. All right, and then, so right now, before we operate the pumps, what's happening is that we've got a bunch of protons. These H pluses are protons. Up here, they're represented by these dots that I'm making. So there's protons all throughout the mitochondria. I know I'm like making a lot right now. I feel like I, again, committed. All right. So there's about an equal, and it might be hard for you guys to see. Let me move it up closer. There's an equal number of dots that I put in the brown area versus the yellow area. Right? So the protons are the same here as they are here. They're like same amount, right? And these H pluses are protons, right? So they're there. What we want to do is we want to pump all the protons out of the yellow and into the brown. That's going against diffusion, against the concentration gradient. So when you're done with it all, well, you get the idea. Something like this. So in order to do that, you have to pump them out with energy because you're going against diffusion. It's the opposite of diffusion. You're going from a lower concentration to a higher concentration. So to do that, I have to, I don't know why I'm writing on this. So I'm going somewhere with this. I'm going to keep writing a few more. So instead of them being equal on both sides, they're all on one side. And to do that, we need pumps. You have to pump it. This is active transport. Active transport needs energy. Usually ATP, but in this case, we're using electrons. So these pumps, this active transport is being used with electrons. The electrons are turning on these pumps, and it's going to pump all the protons into the brown area. So where did we get the electrons? We got it from the Krebs cycle. We're using those electrons to pump all the protons to one side. Now, they're all on one side. They want to diffuse back over. At least half of them do. Right? So they shouldn't have left in the first place, but they were pumped out. Now they want to turn around and come back in. Well, half of them do. Half of them are going to come back in. So that's fine, but you can only come in through here. So they're all going to bottleneck in a few of these places here. You get the idea, right? So we pumped them out. They're going to come back through by diffusion, passive transport. They're coming back through. They're going to diffuse back through just naturally. We pump them out in many places, but you're only coming back through in a few places. So it's like a bottleneck. It's like a dam, like you're damming up water and then you're letting it come through that one turbine to create it. You know, that's what it is. So it's like when you let them through, it's like turning that mill, turning that water wheel that's powering, putting together ADP and P. So you imagine this thing, whatever this purple thing is like a mill, like a factory that makes ATP. How do we turn this factory on? How do we get it going? You got to run water through it, and that like turns the engine. And that's what is going to put ADP and P together to make ATP. That's going to make around 34 ATP. So that is our big ATP payoff. I was kind of making it in understandable terms. You know, technically it's not like a factory. It's making an enzyme work. But anyway, that's the idea. So this was like called co-transport. You had active transport, pumping things against the concentration gradient, you know, against diffusion. And then you had passive transport, which is just regular diffusion. So they're all going to come through here. That's going to, I think I've got like another, and that's what's going to, it's like that. You know, they're all on one side and they're going to come through, and that's like giving power to the ATP factory. And that's where you get the big ATP payoff. So you're getting around 34 ATP from this. You're getting 2 from the Krebs cycle. You're getting 2 from glycolysis. Lysis means what? It's breaking. Glyco, what do you think of?
Glycogen.
Glycogen, that's one thing, because that same word, it means sugar, like breaking sugar. It's in the word glycogen, same root, right? Gen means generate, to create. So glycogen, like making sugar. Here, glycolysis is breaking up sugar. So we get about a total of what? 38, if my math is correct. Something like that. So every time we break up a molecule of glucose, we get somewhere around 38 ATP. Most of it coming from the electron transport chain. This is one of my students. This is one of the students in here. That's fine. Yeah. Does anyone have any questions on this part? Let me go back to that. So break them up. Break the molecule down. Break the molecule down. Turn glucose into pyruvate. Then we're going to turn that into acetyl-CoA. And then the Krebs cycle rips the molecule apart and just harvests electrons. Harvests, if that's the word. Harvests electrons. Then we're going to take all those electrons, NAD, FAD. You'll see them in different forms, right? NADPH, NAD+, NAD whatever. And NAD, FAD, they're carrying the electrons over to here. So they're carrying them from there over to here. And we're going to use those electrons in the electron transport chain. Those electrons are turning on the pumps that are going to pump all those protons out. It's like turning on the pumps here that pump all the water out. If you just let it go, the water's going to keep coming in and it's going to flood us. You have to pump it out.
Pumping the protons out of the mitochondria?
It's pumping it from like one area of the mitochondria to the other. So it's like pumping it from here to here. There's like this outer membrane, this outer space right here. So it's pumping it from this space into here. So they're equal, but we're going to take those and pump them out into here. And then some of them are going to turn around and flow back in again. So it's really what we call what's driving. So ultimately, you want to put, to make ATP, we want to put ADP together with P. So what is the direct energy that puts these two together? It's not the electrons, it's a proton gradient. Meaning all the protons, the H pluses, all the protons are on one side. They're on one side and they all flow back through. But if we were to look at the first law of thermodynamics, we have a proton gradient. No, let's go further. We have ATP. How did the energy in ATP used to be in this, in ADP plus P. We had to put those two together to make this. So here's the energy. Now it's here. It used to be here. How did we get these two together? The proton gradient. So the energy that's in that proton gradient, the fact that they're all on one side, that's what allowed us to put these two together to make this. But where did we get the proton? How could we create that proton gradient? How were we able to put all the H pluses on one side instead of them being like on both sides? we had to pump. We had to turn on these pumps and pump them over. So we used the electrons. So the energy from those electrons was used to pump all those protons over. And then when some of those protons flowed back through, that put the ADP and the P together. Now the energy's here. Where were those electrons before they were harvested? They were in this molecule called acetyl-CoA. Go back to the Krebs cycle here. Of course, before acetyl-CoA, it used to be a molecule called pyruvate. And I'm going to stop here. I mean, as far as like my thing, which used to be glucose. So the energy is inside your blood sugar. Your cell took in that blood sugar and broke that glucose apart into pyruvate. So now the energy that used to be in your blood sugar, which used to be in the biscuit, is now pyruvate. Now there's the energy is now been transferred to acetyl-CoA. It's still there. Krebs cycle rips apart the acetyl-CoA and pulls all the electrons. So there's that energy. It's still there. It's just getting transferred. You just follow all the arrows, and it's a transfer of energy. The energy doesn't get like created or destroyed. It just gets transferred from one thing to another thing to another thing. We stop it at ATP. Or I can even go one step further. ATP, I'm going to use that to make my muscles move. Or contract, actually, right? They contract. And then what happens? I don't know. That's further than what we tend to answer in biology. That's more of a physics question. How you doing? Good. I'm sorry? Steve, oh, yeah, I think she's in that other building. You're always welcome. Nothing going on. Just me talking. You guys can follow us on Discord.
You have a Discord?
No. Just like to say that. It's all on Twitch. But if I did do Discord, do you think people would, like, what would be the best way, like, if I had something that you would actually, like, go in and look? Nothing, huh? The best way is for you guys to set up a GroupMe page and share all the answers. That's what my son does. Both of them. Every class, they're on a GroupMe. Transfer of energy. If you don't get this, okay, don't worry, just erase it. I was just trying to tie everything together. But that was some ideas from that chapter on metabolism. Talk about something else. I have a video that I use the same slides. So, you know, it'll look familiar. Okay. That's kind of a summation of what we've talked about. And you're like, why didn't I put that up before? I forgot I had it. But that's kind of a summation. You know, glucose to 2-pyruvate. We are getting some ATP out of it. We got to convert it into acetyl-CoA. Then it goes through the Krebs cycle, and then the electron transport chain. And that's telling you how many ATP you get around out of a molecule of glucose. But what I really want to talk about is this step, from this step to this step, like going from here to here, right? So when I go from here to here, we need oxygen, because the oxygen has to take away the carbon dioxide, right? And so without oxygen, we can do glycolysis. So we call this aerobic. Let me go to the next slide. Aerobic and anaerobic. You have oxygen available, it's aerobic. If there's no oxygen available, we call it anaerobic. So I'm going to write the words here. AERO. That's aerobic. And then this way, wait. Anaerobic. And then this way is aerobic. So if you have oxygen, we'll continue through. Make the acetyl-CoA, Krebs cycle, blah, blah, blah. Go through the whole process. But what if we don't have enough oxygen? Then for us, we start producing lactate, or maybe a more common word is lactic acid. So why would you not have enough oxygen? Because you're not breathing? No, not that. Like practically speaking. What? No. Stop. Glad I'm not recording this. Oh, yeah, there it is. If you're working out, or let's say you're like running up bleachers, riding a bike up a hill, that's not really a good analogy for you guys, but you know, if you're ever working out and you feel like your legs burn or your muscles burn, like feeling the burn, that's what you're feeling. You're not getting enough oxygen to your muscles to meet the demand of what you're doing. You got to do a tour. Sucks to be you. Good luck, though. They seem okay.
Yeah.
So the anaerobic, it makes lactic acid, or it uses lactic acid?
It makes it because it can't. You have pyruvate, and then you can't convert the pyruvate into acetyl-CoA. There's no oxygen to come and take away that one of the carbons. You can't turn it from like 3 molecules into 2. So it just ferments, and it goes from pyruvate into lactic acid. In the case of yeast, it becomes alcohol, ethanol. Right? But in the case of us, we're working out, and you can't breathe in enough to get oxygen to like your leg muscles to catch up. So then your legs start burning because you're not able to make ATP. Your leg muscles are trying to like let's say you're like running up bleachers or running up stairs, right? You're running up a lot of floors. Your leg muscles are trying to blow through this whole cellular respiration. The cells in your leg muscles are doing like glycolysis, Krebs cycle, electron transport chain. It's doing it like crazy. It's all sped up. But you can't get oxygen to your legs fast enough to keep up. So then you start fermenting the pyruvate instead of going through the cycle. And then that acid burns, right? And then when you stop going upstairs and just rest for a second, the burn will go away really quick. Because then the oxygen gets to your leg muscles and you catch up. Or when they make alcohol, it's the same idea. You dump a bunch of sugar, you got some yeast, and you cut off its oxygen supply. So yeast is kind of like us. It breathes in oxygen. It breathes out carbon dioxide. It consumes sugar, right? But what a yeast will do is turn it into alcohol. So anaerobic means no oxygen available. Aerobic means oxygen available. So if you're doing exercise and you're getting out of breath, like it's hard to, like you can't talk while you're doing the exercise, that's probably an anaerobic exercise. Doesn't mean you can't breathe. It just means you're not meeting your oxygen needs. Yeah. What else? I think I got one more slide. Yeah, it's that one. Oh, why didn't it go? I'm just showing you this last slide, and then we're going to go over stuff on the exam. This you don't have to know. I just put it up there to show you. These are our 3 energy sources from our food: carbohydrates, proteins, lipids. So, proteins are like, meat and beans, and carbs are like sugars that taste sweet and starch. And fats are, fat, olive oil, canola oil, butter, things like that. Your body processes them a little bit different. We talked about the most difficult one, carbohydrates. Carbohydrates has to go through glycolysis. These are the 4 steps. Glycolysis, that's step 1.5. Krebs cycle, electron transport chain. That's what carbohydrates go through. But now look at fats. Some part of fats is made from glycerol. So if you remember, like fats look like the letter E, like an uppercase letter E. The part of the letter E that is like horizontal, those are fatty acids. The part of it that's vertical, that's glycerol. That's not actually a fat. It's just holding the fats. So that you could turn glycerol into sugar. Your body can. But it's got to enter glycolysis. It can't go anywhere else. But the fatty acids, they don't even go through glycolysis. They jump right in at the acetyl-CoA level. Because what your body does is it rips off 2 carbons at a time. If we look at glucose, that's 6 carbons. Pyruvate's 3 carbons. We really want to get it down to 2 carbons long. So these, you can rip them up into 2 carbons and throw them right into acetyl-CoA. Proteins are different. Some of them can go right into the Krebs cycle. You don't even have to make it acetyl-CoA. Some proteins, you can put them right in. They just enter in different places. That's all I was showing you. It doesn't really mean anything, just talking. Oh, look at that. Why does it say NH3 here? That is, if you remember proteins have an amino functional group. I don't know if you remember that, like it's the only one with nitrogen. If you want to turn proteins into energy, you got to get rid of that nitrogen. So NH3 is kind of like ammonia related, right? And so actually the nitrogen part of that protein, if I want to turn this into energy, I've got to rip this off. That means I'm going to I got to get rid of it. I'm going to pee it out, right? So the more proteins you eat, the more nitrogen, NH3s, you're going to rip off of the proteins. The more of those you rip off, the more urine you have to make. The more urine you have to make, the more water you have to add to it. So eating a really high protein diet will, for somebody trying to lose weight, it works in the first week. It'll work. Or second week, right? It works well because it's making you pee more and it's making you lose more water. So what you've actually lost in the first week is a lot of water weight. You didn't lose fat. I mean, all we know is we got on a scale, and the scale reads a lower number than it read before. Oh, I lost 10 pounds. It's not realistic. You didn't lose 10 pounds of what you wanted to lose. You lost 10 pounds of water because you ate so many proteins. But anyway. Let me shut this down and talk about what you want to talk about is the test. So let me see what chapters. Cellular respiration, metabolism. I gotta do mitosis, don't I? I'm gonna put mitosis down here. Before you look at this and say, oh, that's 4 chapters. Okay, it is, but these 3 actually go together because you can't answer questions about this without knowing about these 2. So let's start with the chapter. Well, it's not called mitosis, is it? It's called cell cycle. Let's start with the cell cycle. What would I ask about in the cell cycle? Well, there's 3 parts to the cell cycle. One part is called, can you guys read that? Interphase. And so I would probably want to know what happens in the G part of what happens in interphase. Those 2 things that are happening in interphase. All the organelles are doubling up. And 2, DNA is being doubled up. So in the G phase, that's where the organelles double. And in the S phase, that's where the DNA is made. S stands for synthesis, DNA synthesis. So G phase, organelles. S phase, DNA.
The question to that would be, what is interphase?
What is interphase? Or I could say, what phase is the DNA? Is the DNA doubled up, duplicated, replicated? That would be in the S phase of interphase. So organelles double in the G phase. There's like a G1 and a G2, but that doesn't matter. The DNA doubles in the S phase. So that's one question I could ask about mitosis. Another question I could ask about mitosis is, cell cycle is mitosis. So you remember there were like 5 phases when we talked about it. There was like prophase and prometaphase, metaphase, and I put 1 or 2 things down for each. And so that's all I would ask you. I would say something like what happens in prophase or what happens in metaphase, what happens in anaphase. I'm not going to do all of them. I'll pick 1 or 2. Because that's probably what I'm going to ask you. So for example, for prophase, if you look back to your notes, you'll have something about chromatin condense, right? That would be an answer. For some of them, we only put like 1 thing. Like metaphase, I only put 1 thing.
Those are 3: anaphase, prophase, metaphase.
Yeah, prophase, prometaphase, metaphase, anaphase, telophase. I didn't even put anything for telophase. You know, bacteria don't even have mitosis. They don't go through it because they don't have a nucleus. So they use something called binary fission. It's just like they just split their DNA in half. Like they double up their DNA and they split it in half. And it's like a really super easy process. There's no metaphase or anaphase. So I want you to know that. Prokaryotes, aka bacteria, they go through binary fission. The last thing from that chapter that I might ask you about is what's cancer? Cancer, however you want to word it, it's mitosis. It's unregulated growth of cells. It's like when cells grow and there's nothing to stop them. They just keep going and keep going, and you get it until you get a tumor. Unbridled growth of cells, unregulated growth of cells, too much mitosis without anything to control it. I mean, however you want to answer it, there's like whatever words you want to use. But it's something to that point. The cell cycle keeps going and there's nothing to stop it. And then what does metastasis mean, or metastasize? It means that it's spread. It's spread into the lymph or blood. The lymph's kind of like your blood. It's a way to get things around your body. So once it goes into the lymphatic system or your blood, it could end up anywhere. So I'll probably ask like 3 questions at least about the cell cycle, because we didn't have that one on the quiz. Cell membrane. Then I'll ask you some questions about the cell membrane. You know what? I'm probably not gonna be able to even get to metabolism. But let me talk about cell membrane. Cell membrane is the next chapter. Passive transport versus active transport. Passive transport does not use energy. Active transport uses energy. Active transport goes against the concentration gradient. Meaning against diffusion. So you have to know why. Why would a cell use active transport? It uses it when it wants to go against the concentration gradient. It wants to make something even more crowded. It's going against diffusion. So diffusion makes things less crowded. It goes from a higher concentration to a lower concentration. You're going against, well, diffusion is from a higher concentration to a lower concentration. So if there's more X's outside the cell and fewer X's inside the cell, then things will go inside the cell. It goes to wherever there's less whatever. If there's more water outside the cell and less water in the cell, water moves into the cell. Water goes wherever there's less density.
So diffusion is when a high concentration goes into a lower?
Yeah. That's the whole thing, keeping, it's like keeping airplanes up. You know, if you look at the airplane, like the wing looks something like that, right? So the air is moving across it. The air is going above the wing, and the air is going below the wing. But because of the way that the wing's curved, the air goes faster this way. So because it's going faster, you're creating a high pressure. And the low pressure is all around it. So right above the wing, you notice how it's curved, and even like when you're about to take off, they extend they're called ailerons. They extend it out and make the wing even longer, because you want the air going over the wing to be a lot faster, and it's higher pressure, and the air above the wing, further above the wing's low pressure. So it's lifting. The high pressure wants to go to the low pressure, diffusion. High always goes to low. So that's lifting the airplane. Passive versus active. What's osmosis? Osmosis is the diffusion of water. What is hypertonic versus hypotonic? So hypertonic, more solute. Hypotonic, less solute. So I might say, what is osmosis? Or I might say, describe a hypertonic versus hypotonic. And hyper just means more solute. Hypo means less solute. And then we talked about endocytosis. There was 3 types of endocytosis. It's the last slide that we talked about. It was phagocytosis, that's cell eating. Penocytosis, that's cell drinking. And then receptor-mediated endocytosis. And the key word there is specificity. It's looking for something specific. It's not just eating whatever. It's looking for one particular molecule. I got one more thing from this chapter. I'm not going to do metabolism. I can already see that I can't do metabolism. So it's going to be 3 chapters. The last thing for this, I forgot. Why would a cell use facilitated diffusion? A cell uses facilitated diffusion, meaning that you use a gate, like a door, because, and this is in your notes, it's an ion, like it's charged, or if it's too big, or if you want to move a lot of it at one time. So we're not doing metabolism. We're going to do cell cycle. The thing about with mitosis, the first thing that I talked about, this is the second chapter. The third chapter is going to be what we just talked about. So what should you know from the thing that we just talked about? Where does that? There was three steps to it, right? Glycolysis, Krebs cycle, electron transport chain. Where do they happen? Because the first one happens in step 1 and step 1.5. That happens in the cytosol. Krebs cycle, electron transport chain. Step 2, step 3. That's the mitochondria. Oh. Where does each part occur? And what's the goal? Like, what are you trying to get out of glycolysis? Like, what's the purpose of it? What's the purpose of the Krebs cycle? What's the purpose of the electron transport chain? There's no like strict wording to these answers. Glycolysis, you're trying to make glucose smaller, like get it ready for the Krebs cycle. The Krebs cycle is gonna like harvest the electrons. Electron transport chain, that's the part that makes all the ATP. Or you could answer it in many ways. You could say to create a proton gradient, or to use the electrons from the Krebs cycle. There's like lots of answers. I'm making it kind of a broad question, like what's the goal?
And where does glycolysis take place again?
Cytosol.
And that's the same for acetyl CoA.
Yeah. Because I might ask you what electron goes into the Krebs cycle. What molecule goes into the Krebs cycle? Don't say pyruvate. The molecule that goes into the Krebs cycle is acetyl CoA. What's fermentation? Meaning, like, what happens to pyruvate if there's, what is fermentation? Let me just answer it. It's pyruvate. When it doesn't have oxygen, it becomes lactic acid or lactate. It cannot go further than that. That's a lot of material for you. So you've got actually, we don't have anything from quiz 2 in this, do we? I mean, not as far as direct questions. Because quiz 2 was on cells and I don't remember what it was on. Doesn't really matter, does it? Cells and metabolism. So this is all like new material.
There's a lot of organelles and stuff on quiz number 2.
Yeah, and I mean, the advantage of putting stuff from quiz 2 is that you'd already have it, but it's just more stuff. Like if you didn't get it, I think it's easier just to make it 3 chapters. This is one of the chapters. That's the other chapter. And then the third one I erased. That was mitosis. Now, I keep calling it mitosis. It was also, well, you know, the questions you wrote down. Oh, wow, it's 11:09. Yeah, we gotta wrap it up. All right, I'll take questions, but I'm gonna stop recording here.